You see my screen?
Yeah, yeah, we can see the screen. Yeah.
Okay, good.
All right. So, good evening, everyone, and welcome to the next edition of DTech Dialogues. As promised, we are back with a fantastic new topic of discussion and two brand new speakers, if I may say so. So, let me not keep you away from them. We're going to touch upon some uh, prospects of future therapies with regenerative medicine. So, let me introduce our first speaker for the evening. Uh, we have Dr. Rohit Kulkarni with us, and he's a very known face on the Indian scenario as well. Although he does not live here, so he is an MD PhD holding the Diabetes Research and Wellness Foundation chair position. He's also the section head of islet cell and regenerative biology and associate director of the Diabetes Research Center at Joslin. He's the professor of medicine at Harvard Medical School and associate member of the Broad Institute at MIT, Harvard, and principal faculty at Harvard Stem Cell Institute. He's recognized for excellence in diabetes research by the Ernst Oppenheimer Award from the Endocrine Society, Albert Renault Prize from the European Association for the Study of Diabetes, and many such prestigious awards uh, showered upon him. He's an elected member of the American Association of Clinical Investigation, Association of the American Physicians, and American Association of the Advancement of Science. And his laboratory is interested in investigating RNA modifications, stem cell biology, and organ crosstalk in the long-term goal of finding cures for diabetes and obesity. And I'm telling you, I have heard him and worked closely with him in my past career as well, but he never ceases to amaze me. We also have with us Dr. Nikhil Gandasi. He is the assistant professor of developmental biology and genetics at the Indian Institute of Science, Bangalore, India, and also the adjunct faculty for the Institute of Medical Cell Biology, Uppsala University in Sweden. He's very well published, very active as a PhD guide as well. Several PhD students train under him, and also has received very prestigious awards uh, in his career, including the Avdesha Surolia Award in 2025, the Infosys Young Investigator Award 2024, so on and so forth. Welcome, Dr. Nikhil, and we are glad you are here because we do want to understand what's going on in India as well. With that, moderating this very important session is none other than Dr. Suhas Irande, sir. He is a senior physician and diabetologist, member of the American Diabetes Association, member of the academic RSSDI, and also the honorary physician for the Pune Municipal Corporation. He is very well accomplished and a very senior physician who we all look up to. He serves on the editorial board of JAPI, one of the most widely read scientific journals of India, and has been awarded with several prestigious awards like the Dhanvantri Surya Datta Award of 2023, Legends of Medicine Times Now Award in 2024, so on and so forth. He's also very active on the diabetes awareness front, and we welcome him for this interesting session of DTech Dialogues. And we are, as always, very privileged to have our founders as well, Dr. Jyoti Dev Kesavdev sir, Dr. Manoj Chawla, Dr. Rakesh Parekh, and I'll quickly invite you to, just welcome our guests and say a few words.
Thank you, Purvi. Welcome to Dr. Rohit. He is joining from Boston in the United States. And we are privileged And Dr. Nikki will be sharing, as you said, the Indian experiences. And this is, of course, a new topic for us, and it is aligning with our objective of a DTech. And welcome both of you. Thank you.
Hi, evening everyone. Great to see you all again, and thank you so much, Dr. Rohit and Dr. Nikhil. Hi, we meet again virtually after last time in Bangalore. Looking forward to listening to both of you, and thank you for obliging us with this DTech dialogues that everybody's looking forward to listen to and learn from.
Dr. Rakesh.
Thank you, Purvi, and thank you all the invited speakers for spending your time. I think, Purvi, you can start the session now.
Sure. Dr. Rohit, the stage is all yours.
Thank you, Purvi. Let me first make sure that I'm able to share the slides. I hope you're able to see this.
Yes, perfect.
Great. Thank you so much for the introduction, Purvi. And again, I want to say thanks to you, Dr. Kesavdev, and all the organizers whom I know. I know Dr. Chawla, and also great to meet Dr. Irande and Dr. Parekh. I know Dr. Arvind Sosale and Dr. Banshi Sabu as well. So it's a pleasure to be here. I'm to be here when I say I'm far away. But we've had a snowstorm, and you see we're still digging out, so my face is not yet covered. I just washed off my face with all the snow. But today I'm excited to be there to give you an insight into the topic, which I think would be of interest to DTech Dialogues, and that's about regenerative biology and how we can appreciate what is going on in the context of stem cells and human diabetes. So, as the title mentions, I just want to give you an update on where we are today in the context of how stem cells can be used for therapies. So I'm going to divide my talk into probably like 2 or 3 small components. One is to give you a little bit of the background on stem cells and how we have moved very rapidly over the last 1 or 2 decades to come to the stage we are. The second part will discuss about, what are the successes or relative successes that we have had in the context of treating human diabetes, type 1 especially. And then the last component will merge into the discussion and dialogue part where where we continue to have challenges. So I think that those are the 3 components I want to give you a little bit of insight into. So these are my disclosures. None of this are related to specifically what I'm going to speak today. But just to set the stage for those of you who are not very well versed with this, but the concept of beta cell function and mass, which is plotted on the x-axis. I just want to make sure that you can see my pointer here. Can you see this, Dr. Chawla?
Yes.
Thank you. So beta cell function mass is plotted on the y-axis and age on the x-axis, and you can see that as the human organism grows over a period of time, the beta cell function mass goes up. When the organism becomes obese, the beta cell mass function keeps going up, and that we label as successful compensation. But what happens in type 1 or type 2 diabetes, type 2, for example, is that the compensation fails over a period of time with death or destruction of the beta cells. And the interesting part or the intriguing part of type 1 diabetes is that to begin with, we believe that the beta cell mass function starts at a lower level compared to those individuals who are healthy. And those who are predicted to develop type 1 diabetes always have a reduced beta cell mass, the blue line compared to the red line. And so even though there is no insulin resistance, these individuals have a reduced beta cell mass and function. So the bottom line of all these types of diabetes, including MODY, is that there is a smaller beta cell function and reduced beta cell function and reduced beta cell mass. And therefore, the argument that if you're able to replace these beta cells, then we would be able to cure the disease. So going a little bit more deeper into pancreatic islets and type 1 diabetes, this cartoon here shows a very schematic and very simple concept about the relevance of insulin and glucagon and how autoimmune destruction is a major feature of type 1 diabetes. The images on the right show a wholesome islet at the top, which is shown with immunostaining for hematoxylin and eosin. And you can see that as type 1 diabetes develops, there is infiltration of immune cells which eat up the beta cells and lead to their destruction. So clearly, type 1 diabetes is among the unique diseases which lack beta cells, and an urgent replacement of insulin is one of the requirements. One can look at this also from the Eisenwarth model, which is shown here. Beta cell mass on the y-axis and time on the x-axis. And you can see that we continue to be speculative about the etiology of type 1 diabetes. We believe there is some genetic predisposition, there is some precipitating events, and over a period of time, you would have early, late, and over diabetes. And you can see that all along, the beta cell mass goes down. And we know that all across the world, there are numerous individuals living with type 1 diabetes, and we don't have a cure for the disease yet. So in a nutshell, again, this cartoon and this slide shows the history and progress of type 1 diabetes. I won't go into great detail here, but beginning with 1922, which is shown on the left, when insulin was discovered, and Leonard Thompson received the first injection of insulin in Toronto. We've gone through a number of stages where you have developed long-acting insulin, insulin pumps, CGMs, and here we are in the 21st century where we still continue to struggle to find a cure for the disease. So the last component here is islet therapy. So I want to spend a little bit of time on that for the next few minutes. So we all know that although we are happy that that insulin was discovered by Banding and Best, there continue to be challenges with insulin therapy, as all the physicians on this panel and the listeners are aware, that the blood glucose fluctuation and the injection of insulin are not as well timed as nature has done that for us. And shown here is a cartoon where you have the diurnal glucose profiles in non-diabetic and type 1 diabetic individuals, the type 1 diabetes in the red. And on the right, you can see type 1 diabetes with an insulin pump. You still see that there are issues shown in the red font here: hypoglycemia, lack of tight control, and long-term complications. So all of these issues are a huge economic burden in society. So what about type 2? So I just want to give a little bit of an example of type 2 to argue that even in type 2 diabetes, there's reduced beta cell mass, in spite of insulin resistance being one of the important drivers. So this is one of my favorite slides, because this was done as early as 1955, where McLean and Ogilvy literally weighed the beta cells. So shown here is an image of that paper in diabetes. The upper panel shows those individuals with diabetes on the left and controls on the right. And you see the weight of the beta cells. And you can see that compared to the controls, the patients with diabetes have a reduced number of beta cells. In contrast, the alpha cells are very similar, shown in the lower panel. So this would argue in type 2 diabetes also there is a reduced number of beta cells, which leads to the features of and symptoms of diabetes. And therefore, the argument that for all forms of diabetes, somehow regenerating beta cells in one form or the other is important. Shown here is another cartoon after 1955, 50 years later. Peter Butler and others have shown that the beta cell volume in patients with type 2 is reduced. You can see that obese individuals have reduced beta cell mass compared to lean. But I also want to point out that obese individuals are non-diabetic. In the second bar graph here, show a compensation. So this feeds into the argument that perhaps there are endogenous factors which can promote beta cell growth. But we can discuss that at a later stage. So how can we regenerate beta cells, right? So there are different ways that we can regenerate beta cells. So shown here is a review article from my colleagues Susan Bonnevier and Gordon Weir, where they propose that with data, there's replication of pre-existing beta cells. We could have differentiation of stem or progenitor cells, which have still not been identified in human islets, but it could be coming from ducts or acinar cells. There could be acinar transdifferentiation to beta cells or differentiation in the ductal epithelium. So all these have been suggested to be important in the context of regeneration. What about stem cells, right? So this was something that was started over 2 decades ago when I was still forming my lab in the late 90s and early 2000s, the Joslin Diabetes Center. And I'm sure all of you can recognize these two individuals who received the Nobel Prize for discovery of the stem cells. So John Gurdon, who unfortunately passed away recently, and Shinya Yamanaka, who continues to be an important force in Japan. So here's an example of a human stem cell colony, which is outlined here. You can see that this is how human stem cells look like. So what exactly are stem cells? I won't go into great detail here because many of you are aware of the definition and so on of stem cells. But in a nutshell, you can take a peripheral blood cell and convert them into different types of metabolic cells in the context of having therapy for multiple diseases. So shown here is a cartoon where, for example, one can you have an individual who can donate either a skin biopsy or a blood sample, and we can derive patient-specific iPS cells or induced pluripotent stem cells, which can then be repaired, or they can be differentiated towards the affected cell type and then can be used for disease-specific drug screening. They can be treated with the drug and then put back into the patient. Or alternatively, these iPS cells can be repaired and then differentiated into healthy cells and then transplanted back into the patient. So this is very briefly the concept of how it works. We had an article about how this would be an important holy grail for diabetes over almost 2 decades ago. To argue that this is an important approach to treat type 1 diabetes or type 2 diabetes. And so since then, there have been a number of approaches to generate functional beta cells. What we thought was going to be very difficult has been done by multiple labs across the world, where you can now generate human pancreatic beta cells on the bench using samples from patients. But I just want to also emphasize the challenge that we had and how we overcame it. So at the top is a cartoon showing human pancreas development, which occurs over a period of 9 months, right? This is a well-concerted, amazing development of tissues and cells. And what we have done is to truncate the 9-month conversion into 5 weeks. And you can just imagine the amount of work that has gone into figure out how exactly we can convert an adult mature cell by reprogramming it over a period of 5 weeks, feeding them into distinct growth factors and cocktails of factors to produce beta-like cells. So this is a huge accomplishment, so we should not look down upon this, but we're still not there yet, right? Especially because it's got complex transcription factors which can act as activators and repressors, and we still haven't figured out what exactly is needed for that final maturation of the beta cell, which can be responsive to glucose. So we and others have contributed to optimization of the protocol, and right now we have what is called the Millman protocol, which has been modified by the Kiefer and the Melton protocols. My own lab has contributed to a small portion of this, which I'll be happy to discuss at another time. But suffice to say that there are 7 stages to maturing the beta cells, or I must say almost maturing the beta cells. And so one company, which I'm sure all of you are aware, is the ViaCyte, which has undertaken a huge number of trials. This has happened over at least 10 years. And I'm sure you must have seen this particular publication in 2021 when Brian Shelton was treated with transplanted stem cell derived beta-like cells, and he did become insulin independent. So this was a major success for advances in therapy and curing the disease. 13 of these patients in the first cohort had HbA1c less than 7% after 90 days. But the challenge continued to be that these individuals needed immune suppression. So on the one hand, we are curing some aspects of diabetes, but on the other hand, we're promoting some other issues which are complicating. So I just want to end with the last part here, hypoimmune approach to treat. Where exactly are we here, right? So I think I have another 5 minutes, right, Purvi? So hypoimmune approach to treat type 1 diabetes. Immune cells, we know that attack beta cells and require immune suppression. The solution recently by Sonia Scheffer and colleagues used CRISPR-Cas9 to create what we call as stealth cells. So stealth cells are these cells which can be engineered to lack HLA, human leukocyte antigen class 1 or 2, and that would be required to hide them from adaptive immunity. And you also overexpress CD47, which is an important molecule to prevent macrophage attack. And the long-term survival of this was shown in preclinical studies where non-human primates survived for more than 40 weeks without immune suppression after they received this HIP-edited islets. And in fact, in 2025, the first individual with type 1 diabetes received these engineered islets, and I'm sure you guys have seen this article where just based on N of 1, allogenic beta cells without immune suppression was considered to be a cure for the disease. But this continues to be a challenge still, right? So I think the next series of challenges, this is where I want to end the talk, is so what about the long-term durability? Further studies are required to establish long-term safety, durability, and effectiveness of these cells. How can we scale this up? Right? We can't do this for one individual patient at a time. It costs maybe like $100,000 or $500,000 per patient in this context. Can we develop off-the-shelf stem cells which can produce beta-like cells? So that's the next challenge. What about the complexity, right? So this needs very careful genetic engineering, quality control to be certain that all the cells are completely error-proof. We also need to get regulatory approval for sophisticated cell therapies. And so these are all important hurdles, although we have got successes until now. But I also want to mention that there have been other advances in other disease states, which gives us hope, right? So there are, for MORI-5, for example, we have reported that you're able to model and begin to understand how we can promote therapies for pancreatic hyperplasia. We've also reported that there are other complications in diabetes that can be addressed with stem cells. The other example is macular degeneration can be treated with stem cells. Spinal cord neural stem cells have been shown as long ago as 2018. But I'm sure from 2 days ago, you must have heard about this particular news that came out from Japan, that there are 2 diseases which have now been approved by the Japanese government, Parkinson's disease and heart failure, using stem cell-based therapies. So you can see that we are quickly approaching a stage where some of these amazing developments in stem cell biology and engineering are moving us towards a cure for disease. So soon we would like to address this possibility that you could have this kind of a depot where you go in and ask for a specific stem cell type. I know it's a cartoon, but I think we're closer to this than we were 2 decades ago. But as I continue to mention, there are hurdles, generation adequate numbers, long-term effects, and appropriate delivery of cells continues to be an important hurdle. So we're still far away from stem bucks, as they call it, in terms of trying to derive these cells. So Thank you from snowy Boston, and I think I've kept myself to time, so I'll stop here and move on to the next stage. Thank you, Purvi and everyone else.
Fantastic. Thank you so much. I think that was one of the most fascinating stories of basic biology, and uh, would love to converse more with you on that. Without forgetting, let me mention that it's Dr. Arvind Sosale sir and Dr. Banchi Sabu sir who actually made this happen. So big thank you to them, big shout out to them. And I think Dr. Banchi Sabu is here. I would
Yes,
invite you to
I'm very much here.
would
Bunchi
love
is hiding like the stealth cell that Rohit mentioned. And
Sir, a quick thoughts from you before we move on to the next talk.
Purvi, first of all, thank you. Big shout out for you. You are doing wonderful programs, multiple such program making people aware about DTechCon, which we are going to have in Mumbai. And good news is there are few doctors. Yes, today I got call from them that they want to come to diabetes technology meeting, and I'm sure a lot of doctors who will be joining today online will get benefit of knowing many new things from our today's webinar. Thank you for continuously doing a lot of online scientific activities for our Indian colleagues, doctors. A lot of participants are getting benefit of like basic researchers and scientists like Dr. Kulkarni, Rohit. Otherwise, to get him also very difficult, but we are lucky to have him every year. Once in a year, he comes to India, and we get, we learn from him on once in a year at least in a primer meeting at Chennai or in Bangalore. So thank you very much, Dr. Rohit, for joining us. And
Thank you.
yes, Rohit, thank you for that. Last time we met at where we were, Korea, I think, Korea, in Korean Diabetes Association meeting. Jyoti is there. Thank you, Jyoti. Manoj, Rakesh.
Thank you, Benchipai. Thank you.
Thanks to everyone, those who could join. And now over to Purvi for next session, and then we can have question answer or discussion at the end.
Yes, so we move on to our next star for the evening, Dr. Nikhil from IISc Bangalore. And it's been fascinating interacting with you about this. And I love the session even at Primer earlier this year. So we just can't wait to hear you now. So he's going to talk to us about a single beta cell and insights into type 2 diabetes therapy.
So do you see my screen?
Perfect.
Okay, great. So just minimize this. Okay, so I am going to speak about something which we do every day in the lab, more sort of basic science research, I guess. Just to start off with, I think Rohit did a great job of bringing up all the blood glucose levels during the breakfast, lunch, and supper. I'm going to take the same thing up and then try to say the same thing of how the insulin levels go up. I think all of you organizers would have seen this in the patients. And thanks very much for bringing in the perspective of single cells. Thanks for inviting me over to speak about it. And I'll take advantage to say about each one of these cells in the islets. So in the islets, we have three different types of cells: the beta cells, the alpha cells, and the delta cells. So here you can see even in an islet, although islets are a very small part of the pancreas, right? So it makes up less than 5% of the pancreas. But still, we have predominantly beta cells, which we speak about. And then there's other cells which are, let me try to get this thing pointer. So do you see my pointer?
Yes, we can see it. Yeah.
Great. So you see the green cells there, and then there are blue cells, and then there are also pink cells. And we we do speak about only the green cells, not much about the blue cells and the pink cells there. These are also very important to maintain the blood glucose levels. And this is also important for insulin levels. So that's what I'm going to speak about. So first I'll bring you the, first you bring about the pink cells here. The pink cells are the delta cells that you see there. So these delta cells, although they make up very few, less than 5% of an islet, they play a very important role. So when we speak about delta cells, these are cells which help insulin secretion. So in parallel, what we were wondering when we started this project was, do delta cells do something in terms of type 2 diabetes? First, we wanted to see if the delta cell mass is compromised overall. So when we did that, we did see that when you take a non-diabetic islet, these are human islets, and then you do see with the non-diabetic islets, you see very nice delta cell arrangement that you see there. And with the type 2s, there is a large decrease of delta cells that you see. That's exactly what the cartoon is showing as well. And if you see the relative expression of somatostatin, this has been established before as well. And we also saw the same thing, that the relative expression of somatostatin is also largely decreased. And on top of it, also the number of delta cells is decreased. Is this a cause or a consequence is something which we are dwelling into? We still don't know that. But still, there is a large amount to learn from these delta cells is what we understood at this stage. So then we went on to see what happens with the blue cells that you see here, the glucagon secreting alpha cells. And in this case, to mention and see the effect, we had to label them. To label them, we had to put this in a glucagon promoter, which we successfully did. So we could see these granules in the alpha cells. So here, bear in mind that you're seeing single cells here, where you see these dots. These are the dots of the human islets, where you see the glucagon secreting granules there. And we measure these granules and how much secretion happens through these. And here as well, we do see that there is less amount of glucagon secretion. So this is very surprising to us, because there's actually contradictory studies which do say that there is an increase in glucagon secretion as well. So we went back to this, and we also tried to see which are the patients and how long they have been suffering from diabetes. And we did see that the patients which have been suffering for diabetes for much longer, they are the ones which have less of secretion of glucagon in this case. But this is also very difficult, because the number of the hits that we get with glucagon is very less, because when you try to put in single cells, very few alpha cells actually get retained in this experiment. So with this into perspective, I want to bring back the focus on the islets, where I spoke about these pink cells, the somatostatin secreting delta cells, and the alpha cells which secrete glucagon. So when we look at beta cells there, so where we see, what we see is that we look at these granules, which we look at single cells here. These dark things that you see there, these are insulin granules. Very similar is what we would see in an alpha cell, or in case of delta cells as well, where you see these large dense core vesicles. This is where the single cells in an islet house their hormone inside them. So this is an image of beta cells, but this would be very true in case of alpha cells and also in case of delta cells. So these cells, once they take up glucose, they undergo glycolysis, and then there is depolarization of the cell driven by ATP because of the glucose metabolism, which relates to what happens with the single granules that you see here. These granules fuse to release their content. So these are granules which have packets of insulin that are present. They fuse to the plasma membrane of the cell to release their content. So all these experiments that I spoke about in case of delta cells or alpha cells is very similar to what we did here in case of beta cells. What we do is we take single cells, we then fluorescently label them. So you see these dots that you see there, these black dots. So we label these cells there, and then we try to stimulate with glucose, exactly similar to what happens in our body. And then what happens is something like this. Yes, that's okay. So you see that when you stimulate them, there is this, when the glucose comes in, these fluorescently labeled granules, they fuse with the plasma membrane to release their content, which is in this form insulin. So what our interest has been is to see what happens with these single events that we see here. And once they release insulin into the bloodstream, how they do get replenished, in the sense that the insulin secreting sort of granules, how do they come to the plasma membrane again? How fast can they come? how is this related is what we are interested in. So why is this important? This is important because when you stimulate these single cells and when you see exocytosis, you do see that where there is a large density of these granules. So these are the dots that you see. These are all insulin granules. They fuse to release their content. And in each donor that you see here, these dots correlate with the total number of exocytotic events that you see there on the right there. So these are exocytosis that you see. Exactly similar experiments to what you saw in the previous slide there. And you see a population at the bottom here, which is the type 2 diabetic population. And this is where you see that the density of DOCK granules is less, meaning to say that there is less of insulin exocytosis. So we dwelled deeper into it, trying to see how many of the donors that we saw had density of DOCK granules. These dots that you see there, these are large dense core vesicles, and they do have less numbers in case of type 2 diabetes. This is true in each of the donors that we see here, compared to non-diabetic donors where there is a large number of DOCK granules that you see there, right? So this decrease is what we got interested in case of beta cells. So when we looked at this, so this very much compares to what you see in peripheral blood. You try to see an insulin response, which is routinely done, and you do see a very nice biphasic response, right? So in case of type 2 diabetic individuals, you don't see a biphasic response. What we are trying to do here is to see if we look at these single green cells here, what happens in them. When we look at these single green cells, we do see a very nice biphasic response as well in these single cells. And in case of type 2 diabetes, this exocytosis or release of insulin is completely lost, meaning to say that these single beta cells don't secrete as much insulin, meaning to say this very much translates into the peripheral blood where there is less of biphasic secretion that you see. This goes hand in hand with the number of DOCK granules that I showed you in the previous slide, where the number of dots that you saw in case of type 2 diabetic individuals is decreased, which meaning to say that there is less secretion. So it's something like there is nothing to secrete in the sense that the insulin itself, which has to be there in these granules, are not present in the right place, which means that the secretion in this case is compromised, which is what you see from this graph here, which is decreased massively. So our interest was to see what happens in case of, why is this the case? What is the mechanism behind these single cells? So with this, one of my PhD students, Pallavi, got into something which is glucose transporter. So glucose transporter is a transporter which makes sure that whatever peripheral blood glucose is there, that is taken up by these single cells. These glucose transporters are recruited to the membrane, and these transporters can take up glucose into the beta cells. So she was trying to see if the expression of these transporters is decreased, and that is decreased in fact, and that is shown also by a lot of people from a past decade. But what she went on to see is that the leftover transporters, which are these transporters here, can they recruit to the membrane? Can they recruit to the plasma membrane where the glucose can be taken up, was what she was trying to see. And there, when there is, in the non-diabetic individuals, there is very nice recruitment to the plasma membrane that she sees, whereas in case of type 2 diabetic individuals, that is severely compromised, meaning to say that the less number of exocytotic events or less of secretion that we see from beta cells might be because of less transporters that is recruited to the plasma membrane. That's exactly what is depicted in this cartoon here. So you see that the transporters are very much recruited to the membrane, and that means that there is more and more insulin secretion taking place in the non-diabetic individuals. In case of type 2 diabetic individuals, the transporters are less recruited to the membrane, meaning to say that there is less of insulin exocytosis, which is what I showed in terms of less of biphasic response that I did say in one of the previous slides there, right? So we were trying to use this as a bait to see if we can do anything with respect to small molecules that we were screening. So we screened a number of small molecules to just see. So what we do is we try to culture these cells, culture the beta cells, and we put on small molecules on top, and we try to see if there is increase in glucose uptake, which is what is depicted here, and is there increase in insulin secretion. During this process, we hit upon one of the small molecules, which is FeO4BD. One of the postdocs in my lab, Saptadeepa, did this work. And what she saw was that with FeO4BD, there was more glucose uptake, and then there is more insulin secretion. I have been saying about glucose transporters being recruited to the membrane, and our interest was to see how this recruitment is then seen, or what happens in case of type 2 diabetic individuals where fluophorbidae can be kept longer. So in this case, we didn't do it in human islets. What we did was we did it in case of glucolipotoxicity model, trying to mimic what happens in case of type 2 diabetic individuals. What she saw was that when fluophorbidae was added, the fluophorbidae keeps these transporters much longer at the membrane. And this is what you see in this panel here, where the transporters are rested at a place, which means that glucose uptake is increased, meaning to say she also sees more insulin secretion. We are still wanting to see this in animal experiments to see what happens and what can be delved into it and what we can find out from. So with this, I would like to say that there is biphasic response we do see in case of single cells, and this is driven very much by the glucose transporters taking up glucose and metabolizing them. And this biphasic response is lost even in case of single cells where less of insulin granules fuse with the plasma membrane to release their content. But we do see that with some of the transporters and recruitment of transporters, we can recruit or we can rescue this biphasic response to a certain extent is what we can see there. But there is probably more to do and more number of small molecules that we can screen, and we can also see what happens at a systemic level in case of mouse experiments, and later at a different level is what we see at this point, I think. So with this, we come to a different thing altogether. This is the second part of the talk, where I've been saying about these insulin granules, which are here. And this insulin granules have to really fuse to the plasma membrane to release the content. So this is what forms a complex here. You see the plasma membrane and you see these granules here. They have to fuse to release the content. So a large number of proteins are present in this process. And these proteins being recruited at that place means that this fusion can happen. And this is what made us more interested. We wanted to see what are the different proteins that are present on these glucose sort of insulin granules that are present there, and what are there at the plasma membrane level, and how this can happen that these proteins are present there and they can be pulled more and more toward the cytoplasm, so towards the plasma membrane, so that the fusion can happen. That's what was our question. So we did this with various microscopy approaches, with TIRF microscopy, confocal microscopy, epifluorescence microscopy, etc., where we fluorescently label insulin granules, as you can see here, and we can see various proteins here, in this case syntaxin. So whereas in case of TIRF micrograph, this is what you would see, whereas in confocal microscopy, this is what you would see. In case of, you would see the plasma membrane here, whereas here you see plasma membrane because you're seeing from the bottom there, and that's what you see there, right? So with this approach, we measured a large number of proteins to see how insulin granules are recruited and how these proteins are decorated at these places. So this is a very large pellet. And in this pellet, we wanted to see what acts. And we picked up one of the proteins here, the protein that you see on the right, which is actually very less decorated, which is EPAC2. Why EPAC2? EPAC2 is very interesting protein because this gets recruited to this complex when there is GLP-1 treatment on these single cells. So let's say this is a non-diabetic individual and there's control that you see there, and EPAC is very much not there at all. And when there is GLP-1 treatment, the EPAC very much gets recruited at the plasma membrane, is what you can see there. In case of type 2 diabetes as well, you do see a partial recruitment here. But that doesn't mean that it rescues to the same level as in case of non-diabetic individual. It does rescue at a partial level. So for us, the question was that how does it partially rescue, or how does it not completely rescue? So what we did here is we tried to take many individuals. So you can see HbA1c, which is plotted on the x-axis here. And then we do try to see EPAC2 recruitment, meaning to say that you put on GLP-1 and what is happening to the recruitment, meaning to say how much of this EPAC is recruited to the plasma membrane so that more and more insulin granules can be recruited and more fusion can happen and more insulin can be released. And you can see that as HbA1c increases, there is less and less recruitment of insulin granules to the plasma membrane, meaning to say that EPAC2 in presence of GLP-1 cannot act if the individual is more and more diabetic, is what is being plotted here. So conversely, what we tried was, we tried to develop a mouse model where there is an EPAC2 knockout, and this very much shows that there is an increase in blood glucose, very much similar to a diabetic individual, that you see that the blood glucose level is very high and that doesn't go down at all, is what we see in case of EPAC2 knockouts there. So just trying to see here as well with EPAC2 knockout that there is less and less of insulin granule fusing to the plasma membrane to release their content. And we did see that the dynamics of this fusion also is altered in case of EPAC2 knockouts, meaning to say that in diabetic individuals, although GLP-1 drugs that act on EPAC2 might not completely be doing or the complete thing, Roy dwelled on to the fact that there is this insulin which is secreted through the pumps in case of type 1 diabetes, but maybe in case of type 2 diabetes as well, although there is more and more insulin secretion, but the secretion might not be periodically stimulated, is what I'm trying to say with this experiment here. But we have to see this at a systemic level to confirm the same. So in summary, we do see that in case of beta cells, there is this insulin granules which has to come to the plasma membrane, and then they have to get release ready to get fusion or release of insulin granules. And this is what is lost in case of type 2 diabetes. But in case of increase in GLP-1, what we are trying to do is we are only acting on this step where we are trying to get one step of the granules or insulin granules, which are more and more in case of plasma membrane. So only those can fuse to release their content. But what about the other steps is what we are seeing now, and we want to see what happens with them. Why is this important? This is important because each one of these insulin granules have to be brought to the plasma membrane, and this mechanism should happen for the fusion to happen. So that's why we are looking at various proteins that is depicted here. Why should we do that? We should do that because each one of these proteins have to be recruited for them to fuse to release their content. So with this, I would like to just show you some experiments. I'm just taking time into consideration and trying to skip all these slides here. Sorry for that. And then, yeah, so what we want to do here is that we take these individual cells, individual beta cells, and we try to overexpress these proteins that I showed you in the previous pellets. And what we try to see is that we do see that there is an increase in docking that happens, the increase in number of dock granules that come to the plasma membrane of the cell. We do see syntaxin, we do see RAB3, we do see rafillin. These are our hits. And we do see these as potential targets where we can increase insulin secretion. I did say about glucose transporter a while ago, but these are also the hits that we can hit upon is what we think. Why is this important? This is important because we speak a lot about diabetes risk genes here. So TCF7L2, which is depicted here, or SLC2A2, all these are very well established genes where you do see that when we correlate this with HbA1c, there is more and more anti-correlation that happens. In this case, if there are variants of these genes, then the HbA1c is increased. But surprisingly, some of these proteins that we are looking at, there as well, we do see anti-correlation. And when we try to overexpress those proteins at a systemic level, at an individual cell level, we can increase the insulin secretion. So how can we modulate it with sort of small molecules is something that we are interested in. And that's what we want to see here. And we can see that also at single cells, that these are compromised severely in case of diabetes. So we are trying to see if there are ligands in the cells which can bring more and more insulin granules to the membrane, and there are receptors which can keep them at the membrane so that they can more and more can be there at the membrane at any given time point so that they can undergo fusion. So this is what opens a lot of possibilities is what we think there. And this is what we have been screening through. So my lab works on one of these things, which is more related to single cells and what single cells do and how they can secrete insulin. We also work on exocrine pancreas, we work on pancreatitis, we work on pancreatic aging, where we try to see beta cells and how they respond to aging. We also look at drug targets, which I tried to explain a little bit about. We also look at survival of beta cells. These are different parts what we look into. With this, I would like to thank all the funding agencies and the group which has done all the work. And thank you so much for giving me an opportunity to speak on this. And I would like to take up questions. So thanks a lot.
Great. I think I'm very humbled because to understand how much effort goes into this and what kind of fine research you and your colleagues are doing, I think truly respect that, and waiting for some, more uh, progress and updates for us to use them in the clinic. So I
you.
hand over now the platform to Dr. Suhas Erande sir to take it forward with the Q&A.
Yeah, thank you so much, Purvi, and thank you both the speakers, Dr. Rohit as well as Dr. Nikhil. The dawn of hope is something which I would label both of your lectures as, because the way with which the practitioner handles these situations and the work which you are doing there is something which is certainly a future, I would say. So some of my very simple questions, these are from the point of view of a typical practicing physician or a diabetes specialist in our country. So, Dr. Rohit, excellent presentation. I would ask you a very simple question. And that question is, how do you really qualify a particular type 1 diabetic patient who really needs a simpler therapy is multi-dose insulin? Those who afford the insulin pumps, the advanced ones, they're all right. But how do you really decide that this is one person or this is one type 1 diabetic patient who actually should be qualifying for the advanced therapies, the stem cell therapies of different versions, which you mentioned?
Yeah, that's a great question, Dr. Rande. And as you can see, the question can be debated either way, right? So, but as a rule of thumb, I'm sure all of us will agree as physicians that the individual where the glucose control is so poor and who has one or other complication, especially hypoglycemia, and or the third possibility is that someone who's undergone pancreatectomy, a severe pancreatectomy. So in other words, potentially a person with brittle diabetes, right? A brittle diabetes case, very tough to control with insulin injections, very frequent episodes of hypoglycemia, quality of life is extremely low. Those are the individuals who would classify, at least currently for all the trials, to be on top of the list. And if you look at the YSite therapy roster, you can see that all the 14 patients they chose belong to this category. So I think that would be the first category. But I wanted to extend that discussion a little bit beyond that, because I feel that we should be thinking about how is it going to benefit other individuals with type 1, right? So not only those individuals with brittle, but what about those individuals who just need one injection, who are still relatively okay, but they don't like to be injected, they don't like to be using needles for testing their glucose, especially the children who can't live relatively normal lives compared to others. So I think we, as physicians, we need to extend that argument as to how can we promote stem cell therapy for all individuals with type 1. And maybe one day we would also consider that to include other forms of diabetes.
Absolutely. Because you can always, in fact, you have already gauged that the definition of brittle diabetes itself is rapidly changing with the gadgets which we have to manage type 1 diabetes. So that's very pertinent. The other part of my question to you, Dr. Rohit, is there are other areas which you certainly mentioned apart from the beta cell transplants or the related to type 1 diabetes. But you mentioned about the neuroregeneration, the other parts of diabetes complication, the retinal area especially. I can always tell you that couple of decades ago, we had all India roughly around 150 patients, diabetic foot ulcers, and we had done stem cell therapies for them. The stem cells were derived from the circumcised skins of the different individuals or the same individual, if it is possible. And that was helped or facilitated by the Reliance Research Center in Navi Mumbai. So we had a fair fairly good experience. If the case chosen was very good, we had good results. In fact, quite a few of those patients still are surviving without any additional diabetic foot problems. So what's your experience of using it in non-pancreatic areas?
Yeah, great point. And I think that at least from the viewpoint of the Joslin Diabetes Center, as you mentioned, there have been efforts to consider retinal disorders, which can be devastating to the patient, and whether it's possible for us to generate some stem cell approaches. So there have been one or two examples where Joslin has been part of the system, but these studies were primarily undertaken in Japan, where individuals were injected with stem cells. But I don't think it has gone on to the extent that we expected it, considering the complications of those individuals. But what is more heartening is progress that I just mentioned in the last part of my talk about Parkinson's disease and heart failure. If you look closely at the articles that have come out, and in my own discussions with the investigators who are doing the work, they've developed what they call as a stem cell sheet, and the stem cell sheet can be positioned in the cardiac system, or just over the area where the heart muscle is failing, and it amazingly revascularizes and develops into a syncytium, a cardiac syncytium in that area, to promote appropriate cardiac function. So that is one very exciting development. As you know, cardiac failure is such a major issue in the world. The second one, which is also amazing development, is Parkinson's disease. One of the challenges I mentioned and continue to challenge is how do we deliver these stem cells to the appropriate place? So these individuals were injected under microscopy by surgery into those areas where the dopaminergic neurons are located. And they could show very clearly that just a small fraction of cells which are injected into that area are able to be incorporated without any other undue vascularization to form tumors. And that, I think, is a fascinating development. These are early days, of course, but it's been a year since these studies have been done, and that's why Japan has taken that unusual step of beginning to certify that these approaches can be moved to the general clinic. So those are the two important advances from my perspective that are spearheaded by Yamanaka, who are the person who discovered the reprogramming factors.
Yeah, and a quick response, if I really make also, whether it is a beta cell transplant which you prefer or an islet transplant which you prefer.
Great question, very timely question. I think we're all moving towards needing the whole islet. We know that one cannot ignore glucagon, as Dr. Nikhil also mentioned, somatostatin producing cells. There's a reason why we have this islet, right? I think the hagfish, from where the islet was first discovered, is a very, very old organism. Evolutionarily, the islet has developed into a remarkable cell type or a cell organ, mini organ. All 5 cell types are critical for the beta cell to function well. So I think that many of us are working towards the organoid approach, where we'd like to have clusters, where the clusters contain alpha cells, delta cells, somatostatin producing cells, and epsilon cells. And we'd like to see a day, not in the far future, where we can transplant organoids into these individuals and be rest assured that the glucagon response is static in these individuals.
Thank you so much, Rohit. Purvi, you can interject whenever you like, because you can.
I have one question to ask.
Bansi, please.
Yes. So, Dr. Rohit, first of all, it was a great talk. Since years, last 10, 15 years, every time whenever a patient comes, the first question patient asks her when this stem cell therapy will be available. And recently, in last couple of years, every day on some social media, some Chinese paper comes, there is a cure for type 1 diabetes in China. And then there's some paper which comes that there is a stem cell therapy. In between, in last couple of years, whenever we go to meeting like ADA, Vertex is now a booth, an exhibition area also we see, and Lentidra, which is US FDA approved stem cells, and then there are some clinical trials are going on. So I'm just asking that what answer we should give as a clinician to our patients, that is it in the near future will we able to replace, or will this available, will it become a standard of care for people who are having type 1 diabetes, something like that, or who are insulin deficient? There are people who are not type 1 but maybe severely insulin deficient. Like today AID is, it is there, and we can write and we can tell them this is what we can prescribe you, and probably it will become closed loop also in near future. But do we have similar type of answer for person is seeking an answer that is for type 1 beta cell
Yeah.
replacement is there, stem cell is there, or will it come in a near future as a standard of care? Because since years we are listening, but we are not able to give right answer to our patients.
Yeah, Dr. Sabu, I think that's a very important point, and I also see the dilemma that we all have. And in fact, even I get emails all the time since we do work on stem cells, and from patient, a very concerned parent of a person who's just diagnosed with type 1 in the child is very, very anxious and very, horror-stricken because they have to get insulin injections for the rest of their life. So it continues to be a problem. But I just wanted to step back in history and think about the days when we did not have insulin. So, in the 1900s or late 1800s, they were still trying to figure out where insulin is coming from. So, even at that time, people were wondering whether there would ever be a cure for whether there would ever be a treatment for diabetes. And so, if you look at the 1800s and early 1900s, some of the manuscripts written at that time, there was a lot of anxiety among the physicians, just exactly like what you're suggesting, right? So, the suggestion was, where is it? There is something from insulin, some part of the pancreas. So until 1922, until it was discovered, there was no answer to these patients who just kept coming back to the physicians, even though they said it's much closer, it's much closer. And I also wanted to remind ourselves that Leonard Thompson received injection of insulin only the second time he was cured, he was treated appropriately. The first injection of insulin for Leonard Thompson by Banting and Best was failed because it was not pure. So they had to go back and purify the insulin, and only after the second injection did he recover his glucose levels. So I just make that point that we are at the stage now where stem cells continue to be a challenge, but they're moving in the direction we want it to move. And the studies by Vertex and by the Japanese collaborators are very important. And I did not mention the Chinese advance purposefully because we cannot reproduce some of those data sets. And so we have to be careful where the data is coming from as well. But I wish I could be much more persuasive in my response, but I just want to wait and see because, as Dr. Rande mentioned, we want to look at islet clusters as the therapy rather than beta cells. And I think islet organoids would be the answer for us to address.
Thank you, Rohit, for the excellent elaboration on that. Purvi, have we got time to involve Nikhil now for a couple of things?
We sure do.
So, yeah, Nikhil, actually, I would say that technically, we always gave importance to GLUT4, which is the skeletal muscle GLUT transporter, because we knew that, okay, if you improve that, then or functionality of that, the glucose will be probably utilized better by the muscles and we will have a better control. And exercise is something which can be sort of expected to really improve that. Does it really affect the GLUT1 on the beta cells? Any maneuver like maybe exercise,
So I think
aerobic, anaerobic, whatever.
Right. So the overall thought on GLUT4 and muscle has been more on sort of utilization side, right? More sort of utilizing the insulin, utilizing the glucose. So our what we are trying to say now is maybe that is one part which definitely needs focus. And there is actually many studies to say that GLUT4 function also is compromised severely. That is also because of insulin receptor not being able to take up sense insulin and take up glucose. That is, we are not disputing that at all. We're just trying to say that complementary to that, there is also GLUT2 in the beta cells, which are also probably not responding. So it is just that there's not one thing, but then possibly two things happening at different levels is what we are trying to say there. But I think if we can get something, let's say these small molecules that we're looking at, can we get both GLUT4 and GLUT1 to function at the same time, then it would be really good. But we have not tested that, so can't comment on it.
Absolutely, absolutely. And the other part in which you were delving into a bit of depth, which was that EPAC. So EPAC, as has been, you can say, understood, at least as far as I am concerned, I know that it is important for the chlorination of the insulin granules. So the suitable pH makes advancement of the insulin granules from the maybe in the formation to releasable, readily releasable, etc. Beyond that, you took us to a lot of more functions of EPAC. And I know that EPAC and PKC probably are more related to the recruitment, so to say, and efflux of potassium out of the beta cells, membrane depolarization, etc., or hyperpolarization. So are we having, let us say, any, for example, because some amount of GLPs, they certainly help that EPAC. That's what the literature said. Do you have anything which you are looking up as EPAC function improvement, barring
So
the
now
GLPs?
you mentioned very well about different EPAC functions there. What we are delving into is also with respect to insulin exocytosis, EPAC has many other proteins that it can interact with. EPAC, for one, can interact with a protein called RIM, which is at the plasma membrane. It can also interact with a protein called MUNC13 on the other side. So these are proteins which can still bring them together. So it's very critical that the granule that is there inside the cytoplasm is brought to the membrane. And EPAC is a very soluble protein. It can be either in the cytoplasm or it can be in the plasma membrane. It has both functions. So now our interest has been, how do we pull EPAC more and more to the membrane? So when you put a lot of cyclic AMP, I mean, you put something like forskolin or something like that, which is a hammer, that definitely brings EPAC to the membrane. But that's very artificial. And when we do through GLP-1, how much can we bring it? And what are the, like you rightly said, the pH and how the chlorination happens? That is one part. But can you recruit the right proteins so that the more and more fusion can take place is the other step we are looking at. So in that sense, we do see that with the GLP-1 increase and also the dosage increase, which I didn't speak about. There is also more and more of EPAC being recruited to the membrane, but then there's a saturation level that you reach, and after that, nothing much happens. And that's why I try to bring in the patient-related, HbA1c related stuff there. But I do have a feeling that when we look at EPAC, we are just looking at a part of the iceberg. It's just that we are looking at maybe initial diabetics who are responding to EPAC. We are probably not looking at a different set who are not responding to GLP-1 because their EPAC is not functioning. That is where our interest lies, I think.
Absolutely, absolutely. So the last quick question again to you. There is a school of thought which says that beta cell is the center of the universe. What do you say?
So very well said there, Dr. Renandi. I think the whole thing with beta cells start with this thing that, like, in fact, Professor Rohit also mentioned, it's insulin, and insulin will take care of diabetes. That's what we are made to think like, right? I mean, for decades, we have been doing that. And I think the focus of away from insulin has been shifting from past maybe 2 decades or so because of hypoglycemia that comes with all the things that we do to insulin. I think that center stage has been shifted a little bit. I think, right, like Rohit mentioned very well, that I think the focus should definitely be on the whole islet, and we should not look at insulin only, I think. But the focus remains on insulin because it is all beta cells and insulin, because they go hand in hand, right? So
Absolutely.
glucagon, fasting, all becomes, it's okay. We can manage that, is the thought process there. I think more insight into it, more insight into single cells, alpha cells, will give us ideas into when we treat beta cells, when we treat insulin secretion, what is happening to glucagon secretion. We never look at that, right? This is
Right.
one of the things which is slowly we are going into. But now we say about somatostatin secretion, people will just say, no, that doesn't exist, forget about it. But I think what we are trying to pitch is that, well, somatostatin secretion, in fact, regulates beta cell secretion and insulin secretion. So it's good to look into that as well. So the focus should be on all the cells, is what I would say.
Yeah, I fully appreciate what you said. This should be making the clinicians also more aware of what's going beyond our insulin and other aspects of our management. Purvi, you can please take over. Who
So
maybe?
I just had one question. Dr. Nikhil, you did touch upon the action of GLP-1s on each of these microprocesses. Any further data on, the GIP-GLP receptor agonists, the dual agonists, and are you also collaborating to get more the newer compounds, maybe the ones that are not actually approved, but or trying to see where we can leverage all this information and look at drug targets as well?
So with respect to the newer compounds, there's a lot being done. We have not gotten into the newer compounds yet, but definitely there is a lot of things done with respect to dual agonists with GLP and GIP. So we have been looking into that, and we do see dual effects as well, but not particularly on the mechanism of insulin secretion, just to that effect. But when you look at glucagon secretion, though, we do see that there is a very large extent of change with respect to glucagon secretion. Some of it, especially GLP-1 936, which is the inactive isoform, which is there, that actually sort of inhibits glucagon secretion to a large extent. So there is a scope to look at what happens to alpha cells there at the same time. I think with respect to insulin secretion, the dual agonists play a very good role and they increase insulin secretion. But the opposite effect with glucagon is what is not so encouraging, I think. So we need to look more into that. We have not even looked at delta cells, so that is something to look at, I think.
Fantastic. So I was wondering if we have any thoughts on, because what we see clinically is that actually it's the GIP that is more responsible for the enhanced secretion post, glucose intake or carbohydrate intake. Almost 45% of the insulin is secreted because of that incretin effect, rather than the GLP-1. Do you also see that happening at the molecular level?
So we do see that with respect to insulin secretion definitely happening more and more. We do see that with respect to, not with EPAC2, there is one more protein that is involved, which is MUNC13, which is one of them. And we do see with respect to NSF, there's also one more protein that is recruited. There we do see a change with GIP. So that there are different molecular sort of mechanisms that is putting the focus on GIP and different ones on GLP. I focused on GLP. I didn't focus on GIP. But I think both do put in a lot more force into the cells that there is more and more insulin secretions eventually. But then on the other hand, when we go back to the alpha cells, though, the GIP or the GLP effect, it can be very much with respect to active form of GLP, yes. With GIP as well, there is an effect on alpha cells, but with respect to inactive form, there is a lot more deleterious effect, I think.
Fantastic. Sir, so if we are done, then we can, kind of wind up for the evening.
Thank you so much.
So, anybody else wants to comment or maybe ask questions till the time Nikhil has some minutes with us?
So we had
We
Dr.
really had a
Mithun,
fantastic.
we had Dr. Mithun Bhartia also join in. He's our founder of DTech, but I think he also had another commitment, so he's probably logged off. But once again, I thank you all very much, especially Dr. Arvind Sosale sir and Dr. Banji Sabu sir, who helped us get this webinar together. And a very, very big thank you to you, Dr. Nikhil, as well as Dr. Suhas Erande sir, for being here till late and helping us, disseminate information and learning. And also to Dr. Rohit Kulkarni, sir, I think it's been a challenge with the bad weather, inclement weather there, and yet he managed to log in and be there with us. And again, all our founders, Dr. Banshi Sabu, who's right here, Dr. Jyoti Dev, who logged in earlier, Dr. Manoj, Dr. Amit, Dr. Rakesh, and Dr. Mithun. A very Thank you. And a bigger thank you to RX Events for always helping us technically with everything and ensuring that everything goes smoothly. So, see you all, audience. Thank you for engaging with DTechDialogues. And very soon we will have yet another interesting topic covered. Do send in your questions. We will also email them to our experts, especially today if you're still soaking in all the information and want to, share some ideas or pick their